Good morning, everybody. Thank you for being here for the annual presentations of the Scots Science Scholars Summer Experience. This is our 14th year of doing this, our 14th cohort. We've had the pleasure of working with this great group of students for the past week and a half, I guess, 10 days. And we're really impressed with them, and we know that you are going to be impressed with them. Keep in mind, these folks are not even college students yet. Officially. They start orientation later today, and they have been really busy over the past 10 days doing all kinds of activities, and you're going to hear about just a fraction of that. So as you're listening, first of all, just imagine how busy and tired they probably have been and feel. But also they have been just really enthusiastic group, and we're excited for you to hear from them. My name is Dr. Angelia Gibson. I'm a professor of chemistry here and one of the co-directors of the program, and I am honored to work with Dr. Maria Siopsis, professor of mathematics, also a co-director of the program, and Brett Longwith, who is our STEM success manager. We hired Brett several years ago, one of the best decisions we ever made. We hired him to help with students, and so we call him STEM success manager. But over the years, he has taken on more and more of the work that we do here, and really probably could run this program without us these days. Just does a tremendous job. In fact, I think he gets called the mayor of Sutton Science Center. So yeah, he pretty important guy around here, and so we're honored to work together. We're a great team, and it's a lot of fun to work with them and make this happen every year. We have a lot of people to thank before we get started. And so first of all of this happens because we have amazing support from administration and offices all around the campus. And so I want to shout out just a few folks. Dr. Bryan Coker, he's the president of the college. We're really grateful. He's here today to listen to the presentations, and you're going to hear from him just a little bit before they start. We're really honored to have support from the president's office. Dr. Liz Perry Sizemore, who's the vice president and academic dean, also a tremendous supporter, provides us a lot of guidance for how we operate the program, and her staff. Dr. Ben Stubbs, who is not here with us today, but is a dean of students, and his staff helps with all the things that are not academic that our students are doing. His staff is involved with that, and we especially want to shout out the residence life staff who help us every year figuring out the logistics of getting students moved in earlier than anybody else moves onto campus, and all of those things that go behind the scenes. We're grateful for them. Tyson Murphy and his crew at Mountain Challenge, they help us every year start this program well and get these students doing some things that seem really, really challenging, walking blindfolded through the woods, climbing towers, doing all kinds of things that seem challenging and scary and sometimes even impossible, but they show them that they can do it, and so we're really grateful for them. They also arrange amazing field trip for us that I think you'll hear a little bit about that later on. Want to thank the chairs of the divisions of natural science and mathematics for their support, and then the faculty. You're going to hear about a lot of the faculty who came and taught lessons for these students and helped them learn all kinds of things about science and math. We also there are so many offices, but the ones that we interact with almost every day: physical plant, marketing communications, particularly Wes and Ella, who are our what do we call them? Embedded photographers, embedded press, often this summer, and then staff at all other offices. We're really, really grateful for that. We also want to shout out the folks who really are the backbone for making this happen every year. Our peer mentors. Our peer mentors are just amazing. Every year, this year, every year we think they can't get better, and they just keep getting better. It's crazy. So we have Dewey Whaley, who is a junior engineering major and also a football player here at Maryville College. Chloe Powell, who is a sophomore, biopharmaceutical sciences major and a basketball player here at Maryville College. Callie Condolius, who's a junior environmental science major and a tennis player here at Maryville. And Paul Canios, a junior math and computer science major. Sometimes hard to get a picture of Paul because he's always running everywhere. Paul, if you see someone darting around campus, it's Paul. Cross country, track and field. These guys worked so hard this week. Dr. Siopsis, Brett, and I, we leave at the end of the activities for the day. These guys are here. They're here with them in the evenings. They're here with them over the weekend, coaching them through these presentations, mentoring them, doing the activities with them, taking pictures of them, putting it on social media, all the stuff that you're seeing and all the stuff you're not seeing. That's peer mentors. And we are so grateful to have this team. You all really seriously just amazing. We're very grateful for your support. Very grateful for your help. So thank you so much for being here. I'm going to turn this over to Dr. Bryan Coker, who's going to say just a little bit about the program and about Maryville College, and then Brett will introduce the students.
Yeah, thank you, Dr. Gibson. Great, great to see all of you here. I haven't met you yet. Again, I'm Bryan Coker, president of the college. I'm starting my 7th, tough to believe, year as the president. Pleased to have Dr. Liz Perry Sizemore here, as well as Dr. Geraldine Swan, 2 of my colleagues who help us make everything happen here at the college. We do a lot of great things here. I think some of them we do really well. And this program, in my opinion, is one of those. I believe so strongly in what happens in this program, the results that we see out of this program over the years. Dr. Gibson, Dr. Siopsis, and Dr. Siopsis is wrapping up 2 years now, wrapped up 2 years recently as faculty chair, which means she's been with me every week for 2 years, and we're still talking, and we're still really good friends. I think we became better friends probably as a result of spending all that time together. I'm sorry I didn't get to go kayaking. My life almost right now is focused on trying to get this Alexander Institute, the future science facility, funded and built for all of you and for the future of the college. I think a huge part of our future is going to be in the sciences and in how those sciences connect with this amazing region of which we're part. So what I encourage all of you to do, stick with this program, give it all you've got, make it a part of your overall experience here at the college, and you're going to be a better student for it. You're going to be a better person for it as well. So thank you to Dr. Gibson, Dr. Siopsis, and to Brett. Step out. Where's Brett? Stepped out. Yeah, there you go, Brett. Right there. Okay. And Brett, we can't say enough good things about what Brett does with this program and all he dedicates to it and how he really holds things together. So thank you as well, Brett. I'm part of the fan club. So anyway, congratulations on being part of this. And you didn't come to hear me, so I'm going to be quiet now. So thanks.
Thank you, Dr. Coker. And with that, we can go ahead and have our first group come up. They know who they are. And they're excited to talk to you today. I gotta read it because I changed that. All right, our first group is going to be Aaron, Beckett, and Garrett.
Hello, everyone. My name is Aaron Pomrenke. My intended major is accounting.
My name is Garrett Patterson. My intended major is biology pre-vet. My name is Beckett Lacour. My intended major is engineering.
Before we get started this morning, I would like to give a special thank you to the Department of Education for their post-secondary education, the Office of Tim Burchett, the National Science Foundation, Dr. Gibson, Dr. Siopsis, Brett Longwith, and the peer mentors. So our topic today is navigating the current, kayaking locks, and testing riverwater.
During this presentation, I'm going to be talking about the Fort Loudon Dam, how it works, the purpose of it, pros and cons of it. Then we're going to talk about the lock itself attached to the dam and how it works and how we used it in our kayaking trip. And then we're going to talk about the Little River project and why we did it and how we did it. The dam itself was built in 1940 to 1943 by the TVA. It is used as one of the biggest electrical site in the area, providing some of the cheapest power in the area. And this left corner over here, that is where they make the power using big turbines spinning water constantly. The next purpose is the lock in the right corner. It is used to bring vehicles like barges, boats, kayaks up and down the river or a lake. And then the third reason is for this floodgates here, which provide two of the biggest reasons. One being retail, by that dam being built that flooded the area, allowing for more lake houses, which are higher end properties. And then it also prevents flooding by either opening the gates and letting water out or holding in water when the lower section's flooding. And now the lock is operated by the U.S. Army Corps of Engineers.
The Fort Loudon Dam and lock had a couple of negative effects associated with it. So when it was built, it actually wiped out the town of Old Concord, and it acquired 16,200 acres of land, which relocated 317 families, 60 miles of roadways, and 6 cemeteries had to be moved. It also has some environmental impacts. So the change from a fast-moving river into a deep pool reservoir of a lake, the fish species actually had to change from a species that thrive in the fast-moving water to a species that is more used to a deep pool, like a lake. Also, when the dam releases its water, it changes the flow in the direction in the lake and that depresses the oxygen, which stresses out the native fish and the microinvertebrates.
The lock itself is a one-chamber conventional lock with its main purpose being transporting vehicles up and down. It is actually held together by a miter gate. This gate is a V shape designed by Leonardo da Vinci in 1497. The V shape allows for water on the sides, as you can see over here, to escape around the gate, which means there's less force acting upon the gate, meaning that back in 1497, they actually had humans with levers being able to push it open. And these gates are solid wood or now made of solid steel. And this force, the less force, meaning that motors on the dam were actually able to open it with minimum of like a 1 horsepower. And the less force acting onto the dam actually means that it is easier to use and there's less stress on the motors itself and the doors, meaning that the downtime will be limited and the repair time would also be limited.
So when you get to the lock itself, you'll see a stoplight just like you would on the road. So if it's red, that means it's occupied, you cannot go in. If it's green, that means it's ready for you. The doors will open, you can go in. It also has a horn system, which will, when the horn blows, lets you know that someone is coming out and to get out of the way. Once you get into the lock itself, you'll tie down onto a floating bollard, which keeps you in place so you don't suck down into either side of the gates when it's raising and lowering 70 feet. So the way the floating bollard works is on a vertical track, just like your car would be in a car wash, how it keeps your tire going straight. This bollard is on a similar track where it stays in the same spot the whole entire time. And then while the water is going up or down, the bollard works like your fuel sensor, where it sees the level of your gas. It'll just float right there with you as you're going either, like I said, down 70 feet or up 70 feet.
In this video, you'll be able to see the actual steel gates, which are fully steel and are on both sides. And just looking at the gate too, think how can it open like a piece of paper in water, but it's because of the force being directed from the sides because of its V shape.
It's the bollard moving with us as we're dropping down. It just flows right down on top of the water and stays with us the whole time. And this dam holds roughly 111,000 acre-feet of flood storage that is monitored routinely by water levels, storage percentages, and for quality tested by the TVA. Testing the quality of water is important, whether it is safe for humans and animals to interact with, which leads us to our next project, which is the Little River project.
In this project, one of the biggest reasons is to determine pollution in the water. This is done by finding macroinvertebrates using a technique called cake netting. These bugs in the water that you can find are a great natural and one of the best ways to indicate whether a river or a stream is healthy. The next thing is collecting water samples to monitor the E. coli and coliform levels. This can be impacted by waste plants up the river, or even by human or animal waste. And all this data is being collected to help the argument either against or for the Perry Mills Dam. This dam was created in 1840s, and there's been a large debate for the last few years about whether it benefits or actually hurts the water.
So we actually started off the Little River Project just before Townsend. And we only did 4 water sample sites compared to the normal 11 that Callie would do when she's normally doing the project in the summer. The normal 11 sites would take about 5 hours to do, and we didn't have time for that because we wanted to have an in-depth experience. So the way you start is you take your water bottles and you go to your first sample site, and you'll rinse out each bottle 3 different times, and then fill your first one up and put it into your cooler with ice to be used later in the lab. Then you'll take your lab quest and make sure all of your probes, your pH, temperature, and dissolved oxygen probe is plugged in correctly. And you'll take that over to the river and to your first sample site and put them in the water just enough to where the wires don't get wet so you don't damage the equipment. And you'll wait for the lab quest to balance out on the readings of the temperature, the pH, and the dissolved oxygen, and write those down into your field notebook. Then we did KIGNet sampling. So you took a PVC pipe square that was about 2 by 2, and you put it at the bottom of the river and held it down there. And we took our KIGNet and held it at a 45-degree angle going with the current of the width river. And you had a dancer in the center of the square, and a dancer is someone who kicks up the bottom of the river so you can get all of our material up into the KIGNet for getting those macroinvertebrates. And then after we get the macroinvertebrates into the KIGNet, we'll take it on shore and use tweezers to take the ones that we can visibly see out and put it into the jar to be tested later in the lab.
So now after the field work, we now enter the lab part of this project. So we use 2 machines to determine the health or the damaging water. So first is the Techno machine, which is this one right here. This tells you the ratings of E. coli and coliform. Usually E. coli and coliform is a negative thing when it is seen in the water. The way this works, you take your sampled water from the river, and then you want to pour it in multiple cartridges that has pre-made powder substance. And what you do, you want to shake it up until there's no powder at the bottom. Once that is done, you put it into each capsule, and then you close it. And this roughly takes around 17 hours to fully return your ratings of E. coli and coliform. And like I said, if it rates E. coli and coliform higher, then it will tell you that the water is really not healthy to be around or for animals or humans. Next is the IC, which is this one. This one, the tinier machine, there's multiple vials in it, and there's multiple samples of your water, and it takes roughly up to days to even finish. And this rates your cation concentration and your anion concentration. And next for bug picking, this is from the KIGNet. And what you want to do in the lab part, you have your dish here. Your first step is to fill it up with your distilled water just to a clear, thin layer of it. And then you have your jar. You want to fill this halfway with your 100% ethanol or your alcohol. Then you want to clear it out, and then you obviously have your clicker to count how many macroinvertebrates you find. You have your tweezers and your scupula. So what you want to do, you want to take your scupula and scoop up all the stuff that you have from your KIGNet. Once you have that, you put it in one of the corners, and then you start rummaging through it, because obviously there's rocks, there's leaves, a whole bunch of substances. And what we're looking for is the macroinvertebrates. So with your tweezers, you find all these little tiny macroinvertebrates. And then once you find them, you obviously put your clicker up to indicate how many you have. You put each one into your dish or your jar with the ethanol in. And of course, if you find them in there, it is a clear indicator if it is healthy water or damaged water.
I want to say thank you to the Maryville College Mountain Challenge, the Scots Science Scholars Program, the U.S. Army Corps of Engineers, to the Little River Project, Dr. Duncan, Dr. Unger, Callie, Lily, and Morgan.
All right. Our next group is going to consist of Callie, Kaylee, and Trinity.
My name is Trinity Fletcher, and my intended major here is going to be biopharmaceutical sciences.
My name is Callie Dickison, and my intended major is biology.
My name is Katie Leprich, and my intended major is pre-vet med. Today we're going to talk about a project that we did with our orchards. So we worked on our orchards with Dr. Crane, and then we also did a different lab with Dr. Conkle, used to study the soil health. And we decided we wanted to see how healthy the soil was within our orchards. A little bit background about the orchard is it was started in 2012 by Dr. Crane. It's used for different class experiments. So Dr. Crane uses it and takes students out to count the apples and diameters of the trees to show like human error within studies. And then they pick all the apples and use the apples in the fermentation class to make apple cider. There's two different orchards. There's an upper orchard known as Knowles Orchard, and then there's a lower one called Browns Creek Orchard. Within the orchard, it's all natural growth, so there's no use of pesticides, herbicides, or fungicides. And then they graft the trees, which means like they take a tree, part of a tree, and they put it in with another tree to kind of help replicate trees that they would like. And then all the trees we are talking about today are called enterprise trees, and they were planted in 2014.
Okay, so here's the procedure we did for the orchard lab hosted by Dr. Crane. We gathered the diameter of trees 1 through 7. There were 3 trees in Knowles Orchard and 4 trees in Browns Creek. Once we did that, we went around and we counted the apples on each tree. We had the counters like we talked about in the presentation before us, and we would walk around and we would look at the apples and we would double check each other because we worked in pairs. After that, we compared the data with the class and we used that to find the averages of the number of apples in both orchards as well as the diameters of the trees. After that, we also discussed some confounding variables, which are variables that may affect the outcome of our data. So as we see here, this is a picture of cedar apple rust, which is a disease that can affect the way trees grow. And then right there on the edge is called fire blight, and it's where a tree will look like it's been scorched by fire, and that also can affect sometimes a tree's growth rate. Here's the data we collected from it. So right here, this bar here is for Knoll Orchard, and this is the average number of apples, so it was a little bit above 40. And this one is for Browns Creek, and the average of apples there was way above 60, almost touching 70. So from this data, we concluded that Browns Creek orchards seemed to be more proficient, but we also had to keep in mind that Knoll had more confounding variables, such as a deer that would eat the apples off the trees that was not present in Browns Creek, as well as sometimes more people go to Knoll Orchard as it is more accessible, and sometimes they like to pick the apples off the trees.
So we, in our lab with Dr. Conkle, were testing some of the soil outside of our outdoor classroom here at Sutton. In this, we collected samples from different spots in that. Our group, we had the lawn, so that's what's all over our hands here. But in this lab, we gathered those samples and we did something called the ribbon test. So in the ribbon test, you take a ball of soil, mix it with some distilled water, and you rub it in your hands. That's going to create a log shape, and you pinch it between your fingers. And after that breaks off, you'll measure it. So it breaking off is telling you what kinds of soil or particles are in that soil. So we have sand, silt, and clay. If it's a more clay-like substance, then it's going to have a longer ribbon that comes off of your log. And we'll use that to decide what is more present in our soil.
We also use this sieve on the other side. That top part is gonna take out our gravel, our leaves, grass, stuff like that. The second one is gonna take out the sand, that's our biggest particle, then the silt, and then lastly, you'll be left with clay. Using that, we could, with a scale, determine what kind of soil we had. So these are the multiple you could have. When you're feeling that ribbon test right here, that's part that it broke off. You'll decide what texture it is, if it's gritty, smooth, or if you can't really decide either one of them. So that'll help us figure out what kind of soil our plants are growing in. This is me. I went back out and regathered all of the data that we collected from Dr. Crane's lab. In that lab, he mentioned to us that we all kind of got different answers. There were some groups who got 70 apples on a tree, and that exact same tree, someone counted 120 apples on it. So obviously we couldn't decide if that was some accurate data. So I went back out and I tested each tree for both things 3 times each. And then we put that in a program and found the average of each. And that is how we found this data here. So I was using a caliper, which you can see in this picture, which is measuring the diameter of the trees. And then I used the counter again to count the apples on the tree. We didn't do any of the apples on the ground. So on either orchard, so they were equal. And again, we still had Browns Creek Orchard having the most, but you can see it's definitely different than when we went out the first time. It was almost at 70, and now it's sitting here about 55.
While Callie was going out and counting all the apples and measuring the trees, me and Trinity were working on collecting the soil data from from 4 different sites for each orchard. Some things that we noted with it was we used a square PVC pipes to mark off where we were going to take each site from. And we noted the vegetation so that we could know what kind of grass it was, and then also like if there's clovers or dirt within it. And we used GPS to mark the exact location. And we also used the flags to test the compaction, if there was compaction or not present within each site. Then we used a lab quest seen up here to measure the moisture and the pH of each site. We also gathered a sample about a baseball size of each site of dirt, and we took that back to the lab, and we used that. That's where we did our ribbon test for each site.
While we were trying to find the location for where we were going to take the dirt, obviously one of the big things in experiments is that you want to avoid bias. So as a way to avoid bias, we used a random number generator, and we started 5 steps from the first row of apple trees, and then we counted 24 steps in a big square. And those 4 corners of the square are where we took our samples from. So as you can see, there's a big variance, and this one's a lot in shade, has a lot of dirt. This one has a little bit of dirt and a lot of grass, and some of them had a lot of clovers in it as well. And all of that can affect how the soil reacts in the lab to our tests. We also noted here, this is the location that we got from the GPS. So this first coordinate is the direction from east to west, kind of like your x-axis if you're thinking math. And then this right here is your direction from north to south like this. And by using these 2, you can find a coordinate like you're on a little graph, and that way our experiment was repeatable. So we could go back and we could take from the same site and repeat our experiment from before.
So this is what we found with our ribbon test. In Knoll Orchard, we had a sandy clay loam and a silty clay loam. And in Browns Creek, we had a clay loam, a sandy clay loam, and a silty clay loam.
This was our graph for the moisture that we found from each site. The average in Knoll was about 17%, and the average in Brown was a little over 20%. And we did some research about enterprise trees, and we found that the ideal moisture for them is around, it's important to look at the types of soil too. For this type of soil, sandy clay loams, that a good moisture is about 20%. And so this one is just slightly lower than that. So it could kind of tell us it needs a little bit more moisture, but it's still decently close. And then because Browns Creek has more variance in the soil types, this average being a lot higher was pretty good because we decided that a good range for that should be around 35% moisture. So both of them are a little bit low and they could have more moisture, but overall they both fell in pretty good ranges.
When we did the pH, we took it up each site and averaged it within each orchard. So as you can see, no orchard, it sits at about a 7.2 average. pH level, and in Brown Creek sits about a little bit above a 7. We did some research on what kind of pH level that enterprise trees grow best at, and we found that the 6 to 7 range works well with them, but they prefer the 6.5. So the Browns Creek orchard is closer to that mark, meaning that they have more optimal pH level, meaning indicating that they would grow better.
This is what we've concluded from our experiment. Looking at the evidence that we collected, we concluded that Browns Creek did have the healthiest soil. We also found some really cool things within those two orchards. The thing that we noticed was that Browns Creek produced more apples, but it also had a smaller tree diameter, and that Knoll Orchard didn't produce as many, and it had a larger tree diameter, which was very interesting, especially since the Browns Creek orchard is right beside Browns Creek. So one of our hypotheses that we'd love to go back and figure out is the tree diameter smaller because it doesn't have to hold on to as much moisture at a higher moisture level where it's right there by the river or the creek. So it was able to produce those apples.
Last, we would like to give a special thank you to these people, Dr. Crane, because he gave us permission to go out to his orchards and dig up some of his dirt. Dr. Kunkel for showing us the methods in the soil lab. As well as Dr. Unger for letting us use his room, not only for our lab experiments, but also letting us use some of his tools. We would also give a special thanks to Callie Conzolios for coming out with us and helping us collect our data. Chloe Powell for coming and taking pictures of us on our off time, because who doesn't like getting their picture taken when they're doing cool science things? And as well as all of the other leadership and peer mentors in the Scots Science Scholars program.
All right, here is our second to last group. We have Ellie, Ivy, and Laci.
Hi, my name is Ivy Blanton, and my intended major is nursing.
My name is Ellie Nash, and my intended major is chemistry.
My name is Laci Robinson. My attended major is environmental science and math.
And today we're talking about turtle tracking in invasive species. So to start, an invasive species is a non-native species to an ecosystem which causes harm. They can cause harm in 3 ways, the first being economically, which could be an invasive species taking out a crop yield, overall making a farmer's income not happen. The second is environmentally, which is a non-native species, like taking the resources from a native species and outcompeting them. The last being human damage, which would be an invasive species spreading a disease or parasite causing a pandemic or epidemic. These invasive species typically spread because they are generalist species, meaning that they can live in any habitat and eat any food, which outcompetes native animals. They most frequently spread through human interaction, some examples being the European starling and hogs. Hogs are actually in our Smoky Mountains in Tennessee, and they were introduced as a food source, and now they eat pretty much anything and destroy things, creating havoc in our mountains. Prevention methods are the most important to stop invasive species, which could be making sure transportation methods don't have any species on the bottom of them, and making sure when you purchase animals or plants, you buy from a verified seller. However, since you can't always prevent species, and there's already species in our United States and other areas, we can use chemical and mechanical treatments to stop the spread. Chemical treatments would be pesticides, insecticides, and herbicides, and mechanical treatments would be going and physically removing these plants or animals. Now, the reason I'm talking to you about invasive species is because we have 2 present in our Maryville College woods. The first being kudzu, which is featured here, and the second being the English ivy, which is here. We have these among our 140-acre forest, which is all public and used for recreational activities and fitness. We have a leave no trace policy here, which means whatever you bring, you need to take out of the woods with you. These woods are protected and managed, which benefits both invasive species and native species.
Our eastern box turtle population in our woods, we have a large population in our woods, having around 200 individuals with only around 191 marked at the moment. They are indicator species in our woods, showing that it's beneficial to have them. Since we have such a high population, it shows that our woods also have a very good population of their food sources, being them that they're omnivores. We have not a ghost population since we have juveniles and reproduction happening in our woods, and we have interaction with the English ivy as mentioned as before. The males are found more commonly in the English ivy due to males traveling more often during mating season. This can be seen as a hindrance since males have to crawl over and under the English ivy, blocking their way to get to potential mates. Though it has been found that it is beneficial to the juveniles of the species, being able to hide underneath the low brush. Though the English ivy does cover up the understory and forest floor of our woods, causing choking and suffocation of the native wildlife and plants that are food sources to our animals.
So everything you need to know about an eastern box turtle is that they're native to North America and deciduous forests. They average around 25 to 30 years old, but the oldest is known to be 100, so there's a large standard deviation. Up to age 10, you can see the age of a turtle on its shell due to the rings, but after it hits age 10, the rings can join together because of the small size of the shell, so you aren't able to get an accurate reading. They have a type 3 survivorship curve, meaning when they are first born, a lot die, but the longer they survive, the higher their chances of survival are. They're omnivores, meaning they eat both plants and animals, with their diet consisting mostly of berries and mushrooms and even dead carcasses. Their main predators are raccoons, skunks, snakes, which can prey on the animal, but also mainly the eggs, and they will dig them up. And their other main predator is humans. Their number one cause of death is being hit by a car or being taken by a human as a pet, but humans don't know how to take care of them and they don't make good pets. So most of the time they end up dead. The male and female have a few differences, the first being the color of their eyes. Males will have a reddish color, whereas females have a brownish or yellowish eye color. And the second difference being their plastron, the bottom of their shell. The males will have a slightly angled shell for mating, whereas the females is flat.
Wildlife tracking. So we have a couple different methods that we use to track our turtles. The first one being transect. In our woods, we went out and we chose a section of the woods to scan, which is transect is where we stand in a single file line and we are about arm's length apart and we scan the surrounding woods below us. We're careful to look underneath leaves and logs where that's where they like to hide. And if we find a turtle, which we did on our personal lab out there, we named him Callie, and we collect a bunch of data on them and as much as possible, which we'll talk about later in our next slide. The second one being radio telemetry, which is where we use this device. This is our antenna, which is where the transmission comes through here, our cable, and this is our transmitter box. Using this, we track the tracker on the turtle, and it has a faint beeping noise, which gets louder the closer you get to the turtle. You found a turtle. What's next? First, you chart down as much information as possible. The first one being our turtle number. We use this diagram to give them a tracking number, which, for example, if our turtle was 131, we would put a notch into it right here, here, and here. Next, we put down the GPS location, the date, whether it's male or female. We put its activity, so whether it's resting or if it's active. The vegetation that we found it in, sticks or leaves. We put whether it was in ivy, the invasive species. And then we take its measurements. First one being the carcass, length and width, the plastron, length and width, and whether it's open or closed, because that can affect our measurements. Then we do our shell height, circumference, and mass. Struggles and benefits of tracking. A struggle can be whether the tracker is placed correctly or incorrectly. So if it's placed on the side of the shell, for females, it makes it easier for males to mate with them. And for males, it doesn't really matter if it's on top or the side, but most of the time I do it on top. Another one is that the tracker can detect predators if it is placed directly, acting as a flag. A struggle can be the weather, because rain and thunder can affect whether we can hear the beeping noise coming from this. We also have a specific time window that we can track them, being before 5 p.m. There's also human equipment errors.
Some benefits though is to locate previous found turtles. This can help us find new data, see if they're growing and if they are in any sort of disease or anything like that, being able to make sure the turtles that we have really found are doing well and are growing as should. We can also determine the population health, seeing if we have more individuals or less individuals as last year or the previous year before that. We can see population growth or decline over time, seeing if our population is growing or falling down. We can also do some research to expand our knowledge on turtles, seeing if we have a wider average of turtles that have a different lifespan, or if turtles thrive in different ecosystems. It can also bring awareness to species. Bringing awareness to species is very important, especially if they're endangered or becoming endangered. This awareness can bring more people into studying them, more people trying to develop ways to help them, and more funding towards projects that can benefit them. These are some invasive species in our woods. Our Maryville College has been working hard to get rid of these invasive species that have been affecting our woods. We have actually completely rid of our invasive the honeysuckle bush that are in our woods, now leaving only dead bushes in their wake. This clears up our understory and the English floor layer, our floorware of our forest, letting more of our low-lying natural native plants grow. Though we have been trying to remove kudzu and English ivy using goats during the summer and spring to get rid of the kudzu problem, and using some controlled burns to get rid of our English ivy problem. And why we visualize the data we get. Visualizing can make it easier to share, find, and or easier to sort through. These are the data we have gotten from our turtles that we found on our trip. Cheeky being our radio transect one that we found through the radio. We can see her number, if her care is closed, the different measurements, the shell height, and everything like that. And we can compare it to the new ones we found. The new one we found being Cal, and then MJ and Scotty we found, which were recurring. This makes it easier to sort through and easier to share the information between multiple different scientists. And then we can also use math and Excel to make a model. We, right here in Dr. Siopsis' class, use an invasive species model. We use this model to simulate and predict the pattern of an emerald ash burrow and which way it will travel, and it will travel across the entire forest. This model will show possible outcomes based off the probability within set rules and boundaries. Like in this one, we set the probability to 0.32 within the fact that these are 20 feet apart and the tree will immediately die after getting hit by an English ash burrow. This can show us like what will happen, showing that different probabilities lead to different results, showing that past the 0.5 mark in probability, it almost completely always travels to the cross of the forest. Using this, you can predict outcomes and help us with the Benjamin method. Like in this, we decided we remove this one row right here and leave the gap 40 feet between each tree. It will completely stop the English ash burrow from attacking the rest of the woods.
Thank you to Dr. Unger, Dr. Siopsis, Callie, Brett Longwood, Dr. Gibson, and our few mentors.
Thank you.
Give our other speakers just a second to get settled there. They had some equipment. All right. So here we have our final trio for the day. We have Eli, Kara, and Kenna.
I'm Eli Rowland, my intended major is biology.
I'm Kara Gresham, my intended major is exercise science. I'm Kenna McConnell, and my intended major is biology. This presentation is Beyond the Surface, a closer look at molecular biology through microscopy and electrophoresis with an emphasis on DNA.
So through the Scots Science Scholars program this summer, we have been able to participate in many labs, two of which we chose to focus on, which were led by Dr. Gibson and Dr. Campbell, where we focused on DNA and many laboratory techniques that we use to analyze DNA. We're very excited to show you that in the next few slides.
First lab we'll be focusing on is our microscopy lab. In this lab, we use ultraviolet fluorescence to observe some of our samples. So let's get a quick definition of fluorescence out the way. Fluorescence is a property that causes certain substances to glow when exposed to ultraviolet light. This is done in our case with a solution called cohort we added to some of our samples. This can be used in microscopy to observe cells and details that would escape normal light conditions. This was accomplished in our case with a specialized microscope that could emit ultraviolet light.
So in this lab, we were able to use a light microscope to observe vision niche cells and identify the different stages of a cell cycle based on cell shape and division. By comparing wild type, which were the normalized cells, versus the orb, which were the mutant cells, we were able to observe how mutations influence DNA replication in the cell cycle. We did this by pipetting 1 microliters of orb and wild type each into a test tube. And then we spun that in a centrifuge for 1 minute. The centrifuge was able to remove the excess liquid to the top and leave the dense concentrated cells to the bottom. After that process was done, we then removed that excess liquid to improve the visibility of the concentrated cells that were at the bottom. We then flucked that tube. To get a cloudy substance. Through the microscope, we were able to estimate about 100 cells and make inferences on the cells that were in each cycle of the stage.
All right, so to fully explain what we observed in this lab, we need a little bit of background information on DNA, or deoxyribonucleic acid. So DNA is a double helix structure, which means it's built like a ladder that's been twisted. And the backbone or the sides of the ladder are made out of alternating phosphate and sugar groups that are attached to nitrogen bases or base pairs, which are connected by hydrogen bonds. And the base pairs contain all genetic information that is specific to each individual. And so in this lab, we observed the cell cycle, which is split into 4 major sections. The first one being G1, which is a growth phase. The second being S, which is DNA replication. And then G2, which is the second growth phase. And then M, or mitosis, where the cell actually splits and splits into 2 identical sister cells. In order to make 2 carbon copies of each other, the S stage is probably the most important, which is DNA replication. So nitrogen bases, base pairs are held together by hydrogen bonds, and hydrogen bonds aren't very strong, which allows an enzyme called helicase to unzip them very easily. So you have 1/2 of the strand and the other half the leading and lagging strand. And so because these nitrogen bases, which are adenine, thymine, cytosine, and guanine, have a very specific order, adenine always goes with thymine and cytosine always goes with guanine, it is easy for an enzyme called DNA polymerase to complete the strands. So on half the strand, DNA polymerase reads an adenine, it will create a thymine to match it. So having 1/2 a strand of DNA is sufficient to create an exact copy of the genetic code. So what we observed in our lab is that the orb mutant cells do not replicate DNA correctly, so there was mutations and differences, which means that some cells were shaped rectangularly or rhombus shaped, and they didn't hold the same rod shape that the wild type cells.
The second lab we focused on was a DNA ladder activity. In this lab, we used a process called electrophoresis, which is a laboratory technique used for separation of DNA, RNA, and protein molecules. This was accomplished with an electric current that moved the molecules across a gel, which in itself was accomplished by a buffer solution that contains ions. The samples were stained with a special dye that once they were separated, we could then observe them using ultraviolet light.
So this is our biodiagnosis lab. We used electrophoresis to examine 4 different patient samples of DNA and determine if they were infected with NIQV. NIQV is like a virus and illustrates symptoms of fatigue, aching, fever, like a cold or the flu. We did this by loading 10 microliters from each sample into a gel, and we ran that gel for 30 minutes. After the 30 minutes were over, we photographed the results under UV light. Some of the concepts to understand here were that viral diagnosis include a range of methods to identify its presence, to help detect the virus itself, genetic material in the body's immune response. The value of understanding human cell growth, our DNA, and cell cycle is crucial to how we know a specific virus can impact our bodies.
All right, just a little bit more background information for this one is that in the sample taken for this lab, humans should have approximately 150 base pairs, and to have more would indicate an illness or a mutation. All right, so we observed 4 samples from patients, and the DNA was amplified using a process called PCR, which amplifies specific regions of DNA. In this experiment, the segments would be amplified if this specific DNA was present. So the 150 base pair would be the smallest fragment, and the 250 base pair was influenza, which is a little bit larger, and the 400 base pair is the NIQV, which is the virus that we tested for. The latter provides a scale to the gel, helping to see not only the bands, but how large the DNA fragments are. And pores in the gel act as a sieve, allowing for smaller molecules to travel further down the gel, whereas the larger DNA strands will get caught towards the top. And so here we have our 4 patient samples and then our control on the end. So because we took only human samples, every single one of them should have a line at the 150 base pair mark. And then this one has a second line around 200, which would indicate that this person had influenza or the flu. And this one right here, the DZ, had 1 mark up near the 400, which would indicate that they had in fact had the NIQ virus.
So to wrap it up, DNA is the foundation for holding genetic material that makes us as individuals all uniquely different. By placing an emphasis on DNA structure and buildup, we understand different aspects of ourselves, our health, and the world around us. Combining biodiagnostics and DNA gives us a more visual representation to differentiate between specific viruses more accurately, such as what we analyzed in both of the labs.
We would love to thank Dr. Campbell and Dr. Gibson for hosting the 2 labs we discussed. I'd like to thank Brett Longwith and Chloe Powell for their guidance and mentorship, and Dr. Siopsis and their peer mentors for everything they've done for us during this EdScape experience.
Okay, I have to ask, please, everybody give these kids one more round of applause. So it's pretty obvious from what you saw today that these students already have what they need, right? Some of the things you're seeing today, curiosity. We had students take 2 separate experiences and combine them and do new things with them. For example, in the orchard, we didn't ask them to do that. They just kind of did that on their own. So that curiosity is the mark of a scientist, right? Another thing they did, they were so on task all the time. It was at first when I had them, I thought, is everything okay? Like they were just so focused and so on task. And then we would say, okay, you're going to have to do this extra thing later at STEM Success, which is something we do in the evening. And then they would come and they're like, oh, we did that already. We're ready. We're done. Like, wow. So some of the things you saw today are really some of the hallmarks of what we think a successful Maryville College student will be. However, there's a bunch of things you didn't see today that we saw the last 10 weeks or 10 days, 10 weeks. Well, that would be, I love them, but the last 10 days. Support. I can think of 3 different times where I saw one of these guys, one of these folks stand up next to one of their peers and be supportive. I saw it on the tower. I saw it in the math labs. I saw it when we were setting up and taking down stuff where they were all just, oh, what can I do? I mean, the supportive nature of this group is fantastic. They were helpful to each other and to us and to the peer mentors. So that was also something that we were just floored by. So what's going on? This is kind of, we always talk about community at Maryville College, right? And you guys have built a community here already. You have shown that you can be part of this community and an amazing part of this community, a productive part of this community and a contributing part of this community. So that right there is all we could ask for from you. this is kind of the end of the summer program, but it's also the beginning, right? So what's happening today is you guys are wrapping this up while other families are literally driving to campus to move their first year students in today. So guess what? You're the experts now, right? You've shown that you're part of this community, and you've shown that you are models of the kinds of things we want to see at Maryville College. So what we expect from you now is to go out there and do that thing, meet some new students, expand your circle, share what you've learned here, both lab, math, whatever, how to be in a classroom, but also all those other qualities that were invisible in these presentations, but we saw every single day. If you can take that forward, remember when you look around this room and look at the folks who are here, these are your people, right? These peers that you have, your peer mentors, Brett, us, all the other faculty that are supporting you, right? You have this whole net, and now you can be a net for other people. So we're going to ask you to go forward and take that out into our community. I'm so proud of you. Thank you so much for your great attitudes and your just wonderful being here. And I think we're finished, except we're going to do a final photo with you this afternoon. Thanks to everybody who tuned in. Brett, thank you. Oh, I need to shout out. Dr. Swan is here. Sorry. I just want to shout out to Dr. Swan. She has been so instrumental to this program in lots of ways. When we have written grants, when we have to do grant reporting, she helps with all the data stuff, which is a sort of behind the scenes heavy lift. So I wanted to shout that out. All right, guys, photo, and off you go into the world.